Hello everyone, I'm Sabrina Islam. I'm a second year graduate student in Chemistry and Biochemistry department at the University of Texas at Arlington. Currently, I'm working in Dr. Kevin Shook's lab. Now I'm going to talk about the untargeted analysis of psychedelic and non-psychedelic mushrooms using liquid chromatography quadruple time-of-flight mass spectrometry. Psychedelic mushrooms are also known as magic mushrooms, which contain psychoactive compounds like psilocybin and psilocin and some other tryptamines, which gives effect to treat psychological disorders like depression, anxiety, and PTSD. The purpose of this study is to identify some other potential compounds that are present only in the psilocybin mushrooms, which could be responsible for the entourage effect. We analyze the mushroom in liquid chromatography quadruple time of flight mass spectrometer. We analyze the data in MS-Dial and MetaBioAnalyst, and MS Finder is used to find out the potential compounds from the key features. From this study, some potential key features are found in the psychedelic mushrooms as well as in the non-psychedelic ones. We did untargeted analysis on the samples. This is a method of molecular structure determination in which all ions within a selected M over Z ratio are fragmented and analyzed in a second stage of tandem mass spectrometry by a high resolution mass spectrometer. The samples we have used are both psychedelic mushrooms and non-psychedelic mushrooms. In the sample preparation step, we took 2 to 3 whole dried mushrooms, milled in pulverized 11. Then we dissolved the powder in 0.5% acetic acid in methanol. We did vortex, centrifuge, collected the supernatant. Then we repeated the step C, D, and E again and collected the supernatant, mixed it, and transferred to a LC vial. In the instrumentation, we used Shimadzu UHPLC coupled to a Qt of 9030. Here I have described the LC and MS method. The LC method was 15 minutes long and the injection volume was 5 microliter and we used Raptor biphenyl column. The acquired data were exported from lab solutions and as MZML files and imported into MS-Dial. Data preprocessing like noise setting, baseline correction, peak peaking was performed using MS-Dial. Then the data was imported to MetaBioAnalyst for visualization. In this course plot, we can see that the components in the psychedelic mushroom and the non-psychedelic mushroom differ a lot from each other. From the heat map, we can also see that the ingredients in psychedelic mushroom and the non-psychedelic mushroom vary a lot. We did univariate, multivariate, and machine learning, and we found out 188 key features. The identification of metabolites in untargeted analysis is very challenging since very few MS spectra are available in database. So we used MS Finder. The molecular formulas were predicted in MS Finder from the precursor ion using the accurate mass, isotope ratio, and product ion information. Here I represented only 4 of these compounds which were prevalent in the psychedelic ones, this one in the shiitake mushroom, and this one is in button mushroom and wild mushroom. The future works we want to do are untargeted and targeted analysis with helic and inert coated columns, supervised analysis to investigate compositional differences among psychedelic mushroom strains, and analyzing psychedelic mushroom sample stability. For this study, I want to thank Shimadzu Scientific Instrument, Fridge, and Scottsdale Research Institution. Thank you everyone for having me.